Hi guys, welcome to this video, and today we're going to be going over eukaryotic cells. This is part of A-level biology. Cells are the smallest units that can live independently. Cells contain subcellular structures that each have a distinct and vital importance to the life of the cell. All living organisms are made up of one or more cells. New cells are produced from pre-existing cells via cellular division. So this includes mitosis and meiosis. And that's the overview of cells. Now we're going to dive deeper. Cells are divided into two main types, prokaryotic and eukaryotic. Prokaryotic cells are single-celled organisms, such as bacteria, with no membrane-bound organelles. This means there are no membranes around any of its organelles. Eukaryotic cells make up mainly multi-celled organisms such as plants, animals and fungi. They contain membrane-bound organelles such as the nucleus. They also contain non-membrane-bound organelles, like ribosomes. In this video we will go into further detail on what structures make up eukaryotic cells. First, we're going to do a very quick overview of the differences between plant cells and animal cells, not including their organelles. Firstly, plant cells tend to be larger than animal cells. Plant cells can reach up to 100 micrometers in length, compared to animal cells which can only reach about 30 micrometers in length. Secondly, plant cells are autotrophic and animal cells are heterotrophic. The term autotrophic means that the plant cells can generate their own energy store / food on their own via photosynthesis. In more basic terms, this means they do not need to eat other organisms to survive and get energy. Heterotrophic means that animal cells cannot generate their own energy / food and so must eat other organisms. It is important to recognise that plant cells contain most of the organelles that animal cells contain. However, there are few more organelles that plant cells contain that animal cells do not have any of. First, we will go through all the organelles that both the animal and plant cells have in common. I will let you know when we will go through the structures only found in plant cells or only found in animal cells. Before we start, it's really important to know that some of the organelles we'll go through in this video, you won't need to know too much about yet. For example, the mitochondria and the chloroplast. These are organelles which you need to have a basic understanding of their function, but you do not need to know too much about the structures within them. You will go through these topics in year 13 in more detail. Structures in both animal and plant cells. First, we have the cytoplasm. This is the aqueous structure of the cell, filled with cellular fluid and enzymes. All the organelles part of the cell are contained within the cytoplasm. The cytoplasm is seen to be the site of all chemical reactions in the cell. At GCSE, it might have been described as the jelly-like fluid. However, at A-level, it's much better to say aqueous. This is the cytoplasm on an animal cell and in a plant cell. Next up is the plasma membrane. This is also known as the cell surface membrane. At GCSE, you would have called it just the cell membrane. At A-level, this is not acceptable because many organelles actually have a membrane. So if you just call it the cell membrane, which one do you mean? The plasma membrane surrounds the whole cell. It is selectively permeable to control movement of chemicals in and out of the cell. It compartmentalizes the cell. This means it isolates the reactions from the external environment. It maintains the cell structure and prevents it from collapsing on itself. It contains molecules involved in cell signaling. It contains a phospholipid bilayer made from fatty acid tails and phosphate heads. Don't worry too much about the phospholipid bilayer bit. You will cover this when you get to the topic regarding membranes. Now here's a visual representation of the plasma membrane. Again, don't worry too much about this until you're on the topic regarding membranes. It forms a phospholipid bilayer, which is made from phosphate heads attached to fatty acid tails. The phosphate heads face outwards and the fatty acid tails face inwards. Here is the plasma membrane on an animal cell and on a plant cell. It's important to recognise that the plasma membrane on a plant cell is the inside layer, not the outside. You will look into the plasma membrane in more detail when you reach the membranes topic regarding its structure and roles. And now on to the nucleus. This contains genetic material called DNA, which you're most likely familiar with. DNA codes for all the proteins required for the metabolic activities of the cell. The nucleus is surrounded by a membrane called the nuclear envelope, which contains pores, called nuclear pores, that allow RNA molecules to exit the nucleus. Don't worry too much yet about what RNA molecules are. You will study it when you reach protein synthesis. The nucleolus is an area within the nucleus that is responsible for synthesizing, which means making, ribosomes used in making proteins. Now here I'm showing you the nucleus. Here is the nucleolus, the nuclear envelope, the nuclear pores, and the genetic material, which is DNA or RNA. Here is the nucleus in an animal cell and a plant cell. You will look deeper into the nucleus and its roles when you cover protein synthesis. Now the mitochondria. The mitochondria plays a vital role in aerobic respiration by producing ATP, which provides energy for organisms. The number of mitochondria a cell has reflects its activity levels and energy needs, and so a cell that uses more energy has more mitochondria. The mitochondria is made up of a double membrane. The inner membrane is folded to form structures called cristae. The liquid center of the mitochondria is called the matrix. Mitochondria also have their own DNA and ribosomes used to synthesize enzymes. These enzymes are used in respiration. Here is the mitochondria with its outer membrane, inner membrane, intermembrane space, cristae, mitochondrial DNA, the matrix, and the ribosomes. Here is the mitochondria within an animal cell and a plant cell. You will go into more detail about the mitochondria's roles and structures when you cover the respiration topic. And now we move on to vesicles. Vesicles contain a membrane which surrounds the fluid in its center. They store and transport chemicals and materials. The two types of vesicles you hear about at A level are the transport vesicle and the secretory vesicles. Transport vesicles move materials from one area in a cell to another. Secretory vesicles move materials to the plasma membrane of the cell so that the material can exit the cell. This is called exocytosis. Here is a vesicle that contains the inner fluid and the membrane. Here is a vesicle on an animal cell and a plant cell. You will look over vesicles in the topic regarding protein synthesis. And now we will look at the cytoskeleton. The cytoskeleton is present in the cytoplasm of all eukaryotic cells. It is a network of different fibers that maintain the structure and shape of the cell. All organelles are held in place by the cytoskeleton. When an organelle needs to move, it will be controlled by the cytoskeleton. The cytoskeleton has two main components: microfilaments, which control cell movement and cell contraction, and microtubules, which act as tracks for organelles to move along. Here is part of the cytoskeleton in animals and in plants. It's important to recognise that the cytoskeleton is spread throughout the whole cytoplasm in cells. Now on to the flagella. Flagella are large structures that protrude from a cell. In most cases, the flagellum is used to allow the cell to move, as seen in sperm cells, as the flagella rotate to allow the sperm cell to move to the egg to fertilise it. Here is a sperm cell. As you can see, it has a flagellum. And now we're on to cilia. Cilia are smaller structures that protrude from a cell. Some cilia are stationary and some cilia are used for motility. An example of stationary cilia are those found in the nose. These do not move, but trap any foreign objects within them to prevent it reaching the trachea. An example of mobile cilia is those in the trachea. These beat in a rhythmic pattern to force mucus containing debris back up the trachea away from the lungs. Cilia are made of microtubules arranged in a 9+2 arrangement. There is 2 central microtubules surrounded by 9 pairs of microtubules. These all can move. Here you can see a transverse cross-section of a cilia. This has 9 double microtubules with 2 central microtubules. For example, these are ciliated epithelial cells found in the trachea. As you can see, they contain cilia. This is so that mucus in the trachea can be wafted back up the trachea away from the lungs. You will look into cilia in more detail when you reach the gas exchange topic regarding its roles in the trachea. And now we're on to the rough endoplasmic reticulum, also known as the RER. This is a network of membranes that connect to the nuclear envelope of the nucleus and has a very high surface area. It has ribosomes on its surface and is responsible for protein synthesis. Similar to the RER, we have the SER. This stands for smooth endoplasmic reticulum. This is also a network of membranes connected directly to the nuclear envelope with a high surface area. However, this one is responsible for producing and storing lipids and carbohydrates. As you can see, this is the rough endoplasmic reticulum, with its membranes and ribosomes attached to it. Now this is the smooth endoplasmic reticulum with just the membranes, but no ribosomes. This is the RER on an animal cell and a plant cell, and this is the SER on an animal cell and a plant cell. And now we're going over the Golgi apparatus. The Golgi apparatus, also known as the Golgi body, has a very similar structure to the smooth endoplasmic reticulum. It is a compact structure composed of membranes and does not have ribosomes on the surface of it. Its role is to modify proteins to have different structures and functions, and to then package them into vesicles. Here is a Golgi apparatus with the membranes. This is what a Golgi apparatus looks like in an animal cell and a plant cell. And now ribosomes. Ribosomes can either be free-floating in the cytoplasm or attached to the rough endoplasmic reticulum. Ribosomes are not surrounded by a membrane. This means they are not membrane-bound. Ribosomes are made from RNA molecules. Ribosomes also are made from two subunits. Here is the structure of a ribosome, composed of its large and small subunits. Here are ribosomes in animal cells and in plant cells. You will go into more detail regarding the RER, SER, ribosomes and Golgi apparatus when covering protein synthesis. And now for structures only found in animal cells. First up is lysosomes. Lysosomes are specialised forms of vesicles. They contain hydrolytic enzymes, which are enzymes that break down unwanted materials such as debris and old organelles. They play a vital role in digesting pathogens to prevent disease and also in apoptosis. Apoptosis is programmed cell death. You'll get into that in year 13. Here you can see a lysosome. It contains hydrolytic enzymes. And here's what lysosomes look like in an animal cell. And now we're on to centrioles. In general, centrioles are only located in animal cells. Centrioles are composed of microtubules, which are formed from the cytoskeleton. Centrioles are involved in cellular division. This is what a centriole looks like, and it's composed of microtubules. And these are some centrioles within an animal cell. You will dive deeper into the roles of centrioles when you study the topic of cell division. Now we're going to go over structures in only plant cells. First up is the cell wall. This surrounds the plasma membrane and is composed of cellulose. You will learn more about cellulose when you get to the topic regarding biological molecules. The cell wall makes plant cells a lot more rigid than animal cells, which gives them that rectangular shape instead of a blob. The cell wall protects the whole cell from pathogens and microorganisms as it gives extra strength to the cell. All plant cells have a cellulose cell wall. Here is the cell wall of a plant cell. It's important to recognise that the cell wall is the outer layer, not the inner layer. The inner layer is the plasma membrane. And now we're onto the vacuole. This can also be called the permanent vacuole. Vacuoles exist in the plant cell cytoplasm and are filled with cell sap. Vacuoles maintain the turgor pressure of the cell as it is large and pushes all the cell contents against the cell wall and plasma membrane to give the plant cell its succinct shape. It is surrounded by a membrane called the tonoplast. Here is a vacuole with the tonoplast membrane and a cell sap. Here is the vacuole in a plant cell. And lastly, we're on to the chloroplast. Chloroplasts are found in the green parts of plants, such as the leaves, and not in the roots. Chloroplasts have a double membrane structure similar to mitochondria. They also have a cytoplasm-like fluid called the stroma. Disc-like structures called thylakoids exist in the chloroplast, which contain chlorophyll. They stack to make grana. Grana are connected via membranes called lamellae. Just like mitochondria, the chloroplast contains its own DNA and ribosomes. The chloroplast also contains starch grains produced in photosynthesis. Now here's the structure of the chloroplast, containing the outer membrane, inner membrane, stroma, thylakoid, granum, lamellae, ribosomes, and the starch grains. We rushed through this part because you'll go into a lot more detail about the structure of the chloroplast in year 13 when you study photosynthesis. Like I said, you do not need to know all of this at the start of year 12. You must, however, know all of this by the time you have your year 13 A-level exams. Now, you've heard me talk about membrane-bound organelles. What actually are they? A membrane-bound organelle is an organelle that is surrounded by a phospholipid bilayer, also known as the plasma membrane. You must be able to know which of the organelles we have studied is membrane-bound and which are not. It is important to know that membrane-bound organelles are only present in eukaryotes. We will go over the membrane-bound organelles next. Here are the organelles that are membrane-bound: the nucleus, the rough endoplasmic reticulum and the smooth endoplasmic reticulum, the Golgi apparatus, the vacuoles, this includes vesicles, the mitochondria, the lysosomes, and the chloroplast. The organelles that are not membrane bound include ribosomes, cilia, flagella, centrioles, the cytoskeleton, and the cellulose cell wall. Now that's it for the contents of this video. Try these practice questions / activities to test your knowledge. And here's the answers. Now that's it for this video on eukaryotic cells. I really do hope you found it useful. If you did find it useful, make sure to like this video so it reaches more people, and please subscribe to our channel if you enjoy our content. Make sure to check out our channel because you never know, we might post some content about your other A-level subjects. Bye bye.